All right, so chapter 22. Any questions? What's it over? Oh. On page 200 and what? You're going way back, girl. 820. We have gravity here. Yes? That's why. So that pull, that gravitational pull, and I hope I'm getting your question right. The pull of gravity, the center of the earth, changes the pressure in which things are being pulled towards the center of the earth. So the further away you get from the center of the earth, the lower that pressure is, the pull. Does that make sense?
It does, but why is it the percentages of like nitrogen, oxygen, carbon dioxide, water? What makes those partial pressures change from
that? And then the pull. So think about it. I got a bunch of. Say I have a big magnet, and I have a bunch of little magnets floating around in my room. The closer those magnets are to my big magnet, the faster they're going to be pulled towards that big magnet, right? And when they're moving, that's what creates the pressure, partial pressure. So think of your atmosphere as a beaker, and all of these molecules are moving around and banging up against the sides of the beaker. That's the pressure they exert in your atmosphere. Make sense? So if I'm closer to the magnet, I'm going to move faster and bang into it, higher pressure. Then if I'm further away from the magnet, I'm going to move a little bit slower because the pull isn't as strong. So the atmospheric pressure changes as we go closer to the magnet. That's in the center of the earth.
So your partial pressure of oxygen will decrease.
Correct. As you go further away from the pole.
Sea level is 760.
So
if you go to 440, if you multiply 440 times something, it comes up here.
exactly. And that's going to change within your body too. As things move around, they get away from that pull of the atmosphere a little more. That partial pressure changes as blood flows, as oxygen flows with blood through the system. Does that make sense? She still got that, I don't know what the hell she's talking about, look on her face.
Interactive physiology really is my first one.
Yeah. And don't forget, there's an interactive physiology for the respiratory system too. So think about molecules in motion, and the pressure they exert is just like those same molecules in that beaker. It's when they're in motion and they bang up against the sides. The faster they bang up against the sides, the higher the what? The pressure they exert. So if I take that container, and this is where Henry and Dalton and Boyle sort of all work together to create their theories about gas laws. If I take that same beaker with the same molecules moving at the same rate and I make it smaller, they're going to bang up against the sides faster. What do I do to the pressure in the container? I increase the pressure. But if I make the container bigger, the same molecules moving at the same rate are going to bang up against the sides less. What do I do to the pressure? I decrease it. Make sense? So the atmosphere, you're sitting in a big beaker. Anybody like Stephen King? Big, huge fan. Did you read Under the Dome? No? Awesome book, by the way. If you're bored this summer, it's like, you know, it's one of Stephen King's huge books, but it's a really good read. They're actually making a mini-series out of it, coming in June. I don't know, I saw the commercial yesterday. Very excited. But anyway, it's a story about some mythical or, you know, extraterrestrial force that creates a dome around this town. And nobody can get in and nobody can get out. and then it talks about some of the dynamics of the people. But basically what's happening inside this dome is all of these waste products are building up and building up. Kind of like you guys when you drive your car in our dome of an atmosphere. It's really, really big. It's not like that little town in Stephen King's book. So the waste products aren't going to build up as fast. And some of them might distribute outside our atmosphere. But think of it as a beaker, your atmosphere. And then the center is what? What's the center of your atmosphere in this planet? The center of the Earth. What is the center of the Earth? Do you know? It's a metallic ball of fire, basically. And that has a pull on what's in this atmosphere. That's gravity, right? So when we talk about, and we talked about this way, way back in the semester, we talked about the difference between mass and weight. Do you remember that? Is mass and weight different? Yes. Why?
The mass won't change with the weight.
The particles, the stuff that you're made of doesn't change, but your weight, the pull of you onto this planet, will change the further you get away from what? The center of the Earth. Yes? Same thing with the gases. So higher altitudes have lower partial pressures, and then when we go under the ocean, down deep, what happens to all those partial pressures? They increase. Okay? So, did that make sense? Are we good? You weren't listening to me. I had an amazing explanation. You weren't listening. You'll have to go back and listen. Okay? Question? What's H?
On page 836.
836. What is the H concentration? Let's first of all define that for ourselves. And what does that affect? Say it loud so she can hear you. pH. So hydrogen ion concentration, the pH of your blood, is going to have an effect on your breathing rate. Okay?
But because it says the concentration,
That's you.
and on page 837 it says that hydrogen ions doesn't cross the blood-brain barrier. I'm thinking, how is the concentration in the brain?
Where does it say that?
Under influence of arterial pressure, it says because hydrogen does not cross the blood-brain barrier.
Where are we? Give me a
Paragraph on 837.
second paragraph on 837. Although PCO2, okay. It doesn't say. All right. Although PCO2 and hydrogen ion concentration are interrelated, they're distinct stimuli.
That under influence of arterial pH, because hydrogen does not cross the blood-brain barrier, the increased ventilation that occurs in response to falling arterial pH is mediated through the receptors.
Right.
But if it,
Arterial pH does not influence center. Is that where you are? Changes in arterial pH results?
I was thinking because one side says it's because of the concentration of the hydrogen in the brain, and the other side says hydrogen.
It doesn't say it doesn't pass the blood-brain barrier.
Yes.
It does. It's because hydrogen does not cross the blood-brain barrier, and the increased ventilation that occurs in response to falling arterial pH is mediated through the peripheral chemoreceptors. It's on the first paragraph.
I see it.
Influence in arterial pH.
Okay.
See why I'm confused?
Yeah. Hydrogen does not cross. Well, I know that these, basically what they're trying to explain is that these peripheral chemoreceptors are the ones that are going to control breathing rate. I'm not sure about that whole statement about the hydrogen ions, though. This diagram here shows us where the message comes, and that, remember that cranial nerve that left the family, so to speak, up in the brain, cranial nerve 10? These are where we're going to carry some of that message down that are going to have effects outside the brain region. So blood pH is going to affect those guys and breathing rate and heart rate with the help of that cranial nerve. Let me see, don't stress about that. Let me see if I can somehow come up with a better explanation that makes sense. Because right now, on the spot, doesn't it? Yeah, no, I understand your confusion. So I'll try to clear that up next class. But understand that those concentration of hydrogen ions, the pH, has a great effect on heart rate and breathing rate, hand in hand, because they're constantly being monitored by those centers in the medulla oblongata that are sending message to all of these guys to do their thing based on pH. And pH is going to be affected by, this is another one of those scary diagrams, the concentration of carbon dioxide and oxygen in the blood, which then relates to the concentration of hydrogen ions in the blood. So when we look at this diagram here on page 833, they talk a little bit about influence of carbon dioxide on blood pH. And it has to do with this chemistry that goes on in the diagram inside these little blood cells. This is actually a chemical reaction related to the carrying of respiratory gases. Remember when I said the hemoglobin molecule is kind of like an amusement park ride? How many seats on each hemoglobin molecule for respiratory gas? 4. Okay, and this chemistry is going to help determine who gets on and who gets off and where. So up here, oxygen release and carbon dioxide pickup at the tissue level is described chemically for you. So difference in partial pressures is going to drive the movement, but chemistry is also going to drive the movement of those respiratory gases from one place to another. So because of the difference in partial pressure at the tissue level, and look at the big arrows, the big blue arrows show us that more carbon dioxide, because of the difference in partial pressures, are going to go from a high to a low. We're going to see more carbon dioxide pushing off into the circulatory system here because of that difference. You with me? And what are we going to see oxygen doing? Because of the difference in partial pressure at that level, we're going to be pushing off more what? Oxygen out of the circulatory system into the tissues. The chemistry involved is this little chemical reaction over here. CO2 plus H2O. With the help of an enzyme, because all chemical reactions need enzyme help so they can happen fast enough. The enzyme in question is carbonic anhydrase. Is going to form something called carbonic acid. Carbonic acid is then going to dissociate into what? That little HCO3 is called a bicarbonate ion and hydrogen ions. So if I take that chemical reaction, or any chemical reaction, and I add more stuff to that end, what's going to happen to the reaction? And this is basic chemistry knowledge. It's going to push to the, it's going to push to the right. Yeah? You with me? So if I add a lot more carbon dioxide to this chemical reaction, I'm going to form a lot more what? Acid. So if for some reason And even though carbon dioxide is a waste gas, the balance of carbon dioxide and oxygen is extremely important to help maintain pH. So if I have too much carbon dioxide in my system, I'm going to push this reaction to the right. I'm going to make too much acid, and what's going to happen to my pH? Acid. Oh, go down, right? Which means I have too many what floating around in my circulatory system? Hydrogen ions. Do you see why? If I go the other way and I don't have enough carbon dioxide, what's going to happen to my pH? It's going to go up. It's going to become basic, alkaline. So that balance is extremely important. The chemistry of respiration not only involves the oxygen part of it, but the carbon dioxide part of it too. And both of them work in fine balance to help maintain the perfect pH in your system. What's the perfect pH in your system? 7.35 to 7.45. So in this diagram, we see two different scenarios depending on where we are with respect to partial pressures and the chemistry that goes on. So oxygen release and carbon dioxide pickup in the tissues, we're going to release more oxygen, we're going to pick up more carbon dioxide. And what happens here in the lungs? The opposite happens because why? Because influence of those partial pressure differences, we see that carbon dioxide in the capillary beds in the alveoli is at a higher partial pressure than it is in the alveoli of the lungs, right? So we're going to want to go from a high pressure to a low pressure, big blue arrows pulling out. But the opposite is the case of the partial pressures being exerted by who? Oxygen. More partial pressure in the alveoli, the stuff I breathe in from the atmosphere, than what I have here in the, this is in the circulatory system. So it's going to push out, dissolve in plasma, and then allow an interaction at this chemical level of the reaction. So this is breathing in, right? And then breathing out, we're going to get rid of that carbon dioxide. So I breathe in atmospheric gas in my balloon. We have these differences, and when I breathe out, what am I going to breathe out more of? More CO2. Is there going to be no oxygen in the air that I breathe out? No, it doesn't happen that fast. Give me a break. I can't suck up all the oxygen, right? But there's going to be a higher concentration of carbon dioxide because this will happen in the capillary beds of the alveoli. You with me? So as long as there's air in there, and how has CPR changed from the time that I took CPR, for those of you who may have experienced both? Why? No, that's not the point. The point is there's plenty of oxygen left in that gas, right? So if I take time to stop and do the breathing thing, you're not pushing that blood through the system. So if I do that less, I'm still going to have plenty of oxygen left in that patient's lungs for this to happen. And now I'm continually moving it around. So there's much more heart compression going on than there is me taking time to stop to breathe for the patient. Because statistically, it's not going to make a big difference. We'd rather get that blood, that oxygen, to the tissues. Does that make sense? Make sense to all of you? Yes? So now, before you used to do, what was it, 15 and then 3 and then 15 and 3, and now it's what? Yeah, so you want, again, there's plenty of oxygen left there. I'm not going to suck it all out within that one breath. And we realize that, so that's why that's changed. because we want to get that blood with oxygen to the tissues. And we want to get the blood with carbon dioxide out of the tissues. Okay? So understand, I'm just gonna run through these really quick. Stop me if you don't understand. So we understand atmosphere, yes? Those of you who don't check your email, the lecture is up there. It's been up there for a few days. We should have finished this on Tuesday. We got to move along. So I put both part 1 and part 2 up online for you. Okay? We're going to go on to digestive system today because we got to kick some butt. We're a little behind. Yes, ma'am. Here?
Yes.
Okay.
What makes that difference from the air we breathe to the air that's in our lungs?
What's this?
No, she said CO2.
Are you talking about partial pressure? Just a pull?
No.
Or are you talking about
She's
percentages?
talking about the percentage of
Here.
atmosphere, the approximate percentage of CO2 is 0.04. And if you look at the one in alveoli, it's 5.2. That's a huge difference.
Yes, it is.
She's
Why?
wondering, why does it change?
After my little blood cell explanation, why do you think it changes? I'll go back to the blood cells. Oops, going the wrong way. So I breathe in gas, right? We're down here now. I breathe in the gas. It's got a certain percentage of carbon dioxide in it. There's not tons in the atmosphere, but what am I doing? I'm making it during the chemistry, and I'm doing what to it? Spitting it out into the alveoli, not to the atmosphere, because she asked about why is there such a difference? The only thing that comes to mind, and I'm going to throw it out there, it's like pissing in the ocean when you breathe out, right? Because there's so much atmosphere, and you're just going, dink, with your 6 point whatever percent. Does it increase? Is it measurable? No, because it's so tiny, teeny, that every time you breathe out, you're not going to change the atmosphere, right? Does that help? It was. Well, you're pulling out, you're throwing in more. So in the alveoli, look at oxygen. The hell happened to all the oxygen? See the difference? So this is atmosphere, right? What happened in the alveoli? You sucked it out into what? The circulatory system. So what's left? See, you sucked out. Can you suck it all out? No. And that's the principle behind the change in that CPR method. We still have quite a bit of oxygen left in that gas. So rather than take the time to breathe, keep breathing for the patient, I want to make sure that we push all that blood, keep the circulation going. Can everybody get off the ride at the tissues too? No, just sometimes you ever go on one of those you get caught on there because you're afraid to jump off? And that's what the oxygen saturation curve tells us a little bit about, is we're not going to dump off every single tiny, teeny bit of oxygen off of every single molecule every time we have this chemistry exchange, but the majority is going to come off. Enough so that everybody gets what they need. So when we look at this, are you guys following me, or am I making no sense whatsoever? Who says you're making no sense whatsoever? Okay, good. So this diagram shows us the difference in pressures, okay, and tells us a little bit about why we're going to push things off or pull things in with respect to respiratory gases. So Dalton says what? And he's on page Dalton, Henry, and Boyle. Dalton says what? No, that's Boyle. Dalton talks about each of those specific gases and their separate individual partial pressures. The sum of them all creates the total. So take the same beaker, put oxygen, nitrogen, carbon dioxide in the beaker. It's like putting myself, a real fast runner, and a real slow runner in the beaker and telling us to run as fast as we can until we smash into the wall. Yes? Are we all going to be running at the same speed? No. Do all those gases exert the same partial pressure? No. But all of them together are going to create the pressure in the atmosphere. Yes? That's Dalton. What does Henry say? Henry says all of that pressure is related to how fast or how slow things can do what? Dissolve in liquid. Gas to liquid. Did anybody go out today? Anybody near water? It's really cold outside. What was hanging on top of the water? As the breeze blows across, we get a lot of what? Yeah, the vapor-like. What is that? Yeah. Gas going, and here's the chemistry part, gas going from a liquid to a gas, or gas coming from the atmosphere into a liquid. So Henry talks a little bit about how those pressures relate to how fast something's going to dissolve or dissociate gas-wise. And then Boyle talks about the container thing. So the three of them all together are what we call the gas laws. How fast or how slow something is going to go into the blood or be departed from the blood. And all three of these principles are related to that. We good with all three guys? So basic definition of their explanation of how gases act. Understand this. Understand that the composition of alveolar gas is going to be different than the composition of the gas in the atmosphere because I'm pulling stuff out and throwing stuff back in. So I hope we covered that. We're good with that? Okay, and who's going to drive that? Concentration and who else? Big who else? Who's going to drive the partial pressures, the pressures? You always think high to low. So, also...
That's why it's hard to breathe it way up because of that reason.
Correct, because remember, I have to create the pressure gradient, right? If I don't create that high to low difference, then gas is going to stay put. I'm not going anywhere. I don't want to stay where I am unless you create a lower pressure for me, right? So when you go higher, what do you have to create? Yeah, a bigger gradient. So in the beginning, sometimes you're breathing.
These people that are conditioning for that type of stuff, do they acclimate? They have to go through like constant ins and outs and like.
Your body will acclimate to the situation it's in. When I change drastically from one to the other, you're going to feel it. You're going to be more aware of the changes that are going on to acclimate. But once you're in it for a while and your respiratory rate evens out and the rate at which you produce red blood cells and carry oxygen evens out, you're no longer going to be aware of that anymore. Does that make sense? And if you do it slowly, you probably never will even notice it. because your body will take care of all that for you. So those centers in the brain are constantly monitoring the oxygen levels, carbon dioxide levels, via the pH. So alveolar gas, thickness and surface area of the respiratory membrane. So if there's too much fluid floating around in the alveoli, that's the respiratory membrane, is the gas exchange going to happen efficiently? So what happens when you have pneumonia? Why is that a big problem? So I get off the ride, and then I have to go swim through nastiness to get to where I want to go. Does that happen fast? No. So the thicker the membrane, or if there's fluid that's going to slow things down in the membrane, I'm going to start to get into some trouble. And that's where the discussion of perfusion happens. So basically the rate in which things move during the chemistry that I discussed with the cells. So, everybody good with that? So some of the different influences. Also, if I mess with the pressure, I can also influence perfusion as well. If I change pressures, I'm not going to be able to, again, go with that gradient. So internal respiration is discussed, talking about that exchange that happens at the cellular level, at the tissue level. Oxygen transport, we know it's a hemoglobin molecule, right? And we know there's 4 seats on the ride, and I can carry carbon dioxide or I can carry oxygen on the ride. Yep. 4 seats, 4 different seats. Doesn't matter who's in them. Yes? The affinity, and that's where the word affinity for under those saturation curves, it's that attraction between the molecules and the site, that heme portion of the molecule where the molecule is attracted to sit. We have an attraction for both carbon dioxide and oxygen. The release is also extremely important. If the affinity is too strong for that seat, that heme molecule, the exchange of gases, the get off the seat and go to where I need to go, is not going to happen. And that's what they talk about in the oxygen saturation curve. So some of the factors that are going to influence me getting off the molecule are things like temperature and pH. Okay? And then pressure. Carbon monoxide sits on the seat and buckles in. Not getting out.
It's actually more, the heme is actually contracted.
Correct. Correct. So carbon monoxide is an issue with the oxygen carbon dioxide saturation curve, because once that molecule is introduced, it's going to park itself in that spot for a much longer time, because carbon monoxide has a very high affinity to the heme portion of the molecule. So again, my analogy with the amusement park ride, it's like carbon monoxide sits in the seat and buckles in and is not going to leave. Is that a problem? Huh? How come? Because nobody else is getting on the ride. No exchange. Eventually, all of that carbon dioxide is going to build up. Not enough oxygen is going to lead to a decrease in ATP production, and you die. Oh, do we breathe in carbon monoxide? Yeah, it's in our atmosphere. Especially if you're hanging out, like, you know, in the parking lot and walking by somebody's tailpipe. You're breathing it in. Yeah. But it's not going to be the majority, again, of your molecules that you breathe in. Do you have some sites that are blocked up by carbon monoxide? Yes, you do. But eventually they'll get off. Make sense? But if most of your atmosphere is, if you're breathing in the air from the tailpipe, for long periods of time, then all your rides are going to be full. Does that make sense? All right, association of oxygen and hemoglobin then is what the discussion is as far as getting on and getting off that hemoglobin ride. And some of the influences are going to be that partial pressure. Other factors are going to be pH. And again, we saw how closely pH was going to be affected by the actual concentrations of oxygen and carbon dioxide in the system. A couple of terms you should know are things like hypoxia. What's hypoxia? Yeah, low oxygen levels. On page 829 in your textbook under homeostatic imbalances of the respiratory system, we see a bunch of terms. You should just have a basic definition under your belt for. So anemic hypoxia, not getting enough oxygen to where it needs to go because why? I don't have enough rides. I don't have enough hemoglobin molecules. Anemia. Ischemia or stagnant hypoxia. What's that? And this is where the new method of CPR comes in. If the blood isn't moving, we have an issue. Yeah, so if we block movement, we're going to have trouble with hypoxia. And the rest you guys can read real well, so basic definition 4. Keep in mind that reaction on the other side of page 829. So carbon dioxide plus water is going to lead to a molecule called carbonic acid. And then that dissociates into hydrogen and bicarbonate ions. And the two in balance are going to help with transport of respiratory gases. If I put too much on one side or not enough on one side, I'm going to affect what? pH. So anybody ever hyperventilate? What happens? Who monitors your respiratory rate? The medulla oblongata, yes? And how does it do that? Based on what? pH, 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 pH. So if I get rid of too much carbon dioxide from this reaction, that means I'm not making enough what? Acid. So what happens to my pH? Goes up. What's that called? Alkalosis. And if it happens because of the respiratory system, it's called respiratory alkalosis. Yes? The opposite can happen. So what do they make you do when you're hyperventilating? You get a bag and you breathe in. You keep breathing the same air in. How come? Remember, when you breathe out, there's still plenty of oxygen when you breathe out, but the concentration of carbon dioxide increases. So as you breathe in that air with a little bit of a higher concentration of carbon dioxide, what are you going to do to your pH? Bring it back into sync. More carbon dioxide, more acid, bring down the pH to where it should be. And then what happens to your breathing rate? It slows down. Yes? Makes sense? So pay attention to that reaction. Understand what it means. And when you look at the blood cells, you'll see that that will make more sense. So why is this lady sitting around, these guys on the beach, these guys in the ocean? It talks about the partial pressure of gases at different levels on our planet. Do we understand that at sea level, that's the 780 millimeters of mercury? If I go lower, what happens? The pressure increases. At high altitudes, it decreases. So less O2 at that partial pressure. Does it mean there's less oxygen in the atmosphere? No! It just means what? It's not exerting as high a pressure. Remember the pressure. Correct. So remember, I'm going to go from a high pressure to a low pressure. Do you see why it would be a problem if I immediately go to that high altitude? My body will adjust if I slowly, in increments, in either way, change atmospheric pressure. We'll adjust to that. But if I do a quick change, then I'm going to have some problems.
You say that I had some friends that moved to Colorado. He said that when they moved from Maine to Colorado, when they lived at a really high elevation. They actually said while they were there for the first month, it was
Kind of lightheaded.
Yeah.
But then they adjust, right? And that's why, what's that basketball team? They always seem to have a little bit of a edge when we go and play them when they're in Colorado. Who? Anybody? Basketball team, Colorado. High elevation. Denver Nuggets? Yeah? Yeah. They seem to have a little bit of an edge sometimes when we go away. Yes? How come? Because they're all acclimated to their high pressure atmosphere, and all the other basketball guys are like,
Give me your bag.
right? So this is an oxygen dissociation curve. Remember, I have to pick it up, get on the ride, and then I have to get off the ride. Yes? If I don't get off the ride, we have some issues. So this talks about some of the different factors that affect me getting on and getting off the ride. That's what they describe under the Haldane effect, and some of the influences of carbon dioxide on blood pH and chlorine shift and all of that stuff has to do with this dissociation curve. So when I look at this data, normal body temperature is going to allow for a certain number of oxygen molecules to get off the ride. What's normal body temperature? 98.6 degrees Fahrenheit, 37 degrees Celsius. Yes? So they have 38 there. That's kind of an average-ish. And body temperature. So what happens when body temperature goes too high? Versus body temperature goes too low. So temperature also influences dissociation. Do more get off the ride if it's too high? Yeah. How about too low?
No.
Not enough get off the ride. You see the difference?
Is that what happens during hypothermia?
Yeah. You also get hypoxia as well. Because a lot of your molecules aren't leaving because of this dissociation event. How about down here? This is what? Describing what? pH, the other influence on dissociation. So normal, 7.35 to 7.45, dissociation curve here. What happens when pH does what? And it's not by a lot. What's this?
It's more acidic.
More acidic, right? Because what? Too much carbon dioxide and making too much carbonic acid. It's dissociating into hydrogen and bicarbonate. Yes? That's not a big jump in pH, is it? So if I go to 7.2, look at how I've changed who's going to get off the ride versus this pH. Again, not a big jump. But what happens here? When I decrease carbon dioxide, I don't have enough. pH goes up. What happens to the dissociation curve? I'm not getting off the ride. Yes? So the two work together to help carry oxygen, carbon dioxide, and let it off or dissociate it from the molecule. Did you have a question? No?
So, if pH was 8, that would be when you would
If the pH was 8, you would die. But you wouldn't have what? Think about your receptors and what they're monitoring for. So if the pH was 8, what would that do to your breathing rate? You would start to quicken your breathing rate because what your body want to do. Yep. What's your body want to do?
It wants to take the balance.
Yeah, it's trying to get what? More.
CO2.
Right. If you have too much CO2, what's going to happen to your breathing rate? Hypoventilate. You're going to slow down your breathing rate. Yes?
Now, the only way to...
Well, your slowed breathing rate is going to result in too much carbon dioxide.
Okay. I was going to ask a question, but just answer.
Okay. Does that make sense? Okay. So that's what they talk about under pH and temperature influences. We already talked about our cute little cells and the chemistry that goes on. Then they talk about neural mechanisms. So those centers in the medulla oblongata, respiratory centers, consists of nuclei and axons that are going to control the muscles that control respiration. Everybody knows what they are, right? What are the muscles that control respiration? The intercostals and the diaphragm. Okay, so we see that those guys are going to either speed up muscle contraction or slow down muscle contraction based on some of the other factors we've discussed. So when you talk about neural mechanisms, you should know the respiratory centers, the pontine respiratory center, then we have the ventral respiratory group, and again, they're going to just signal different people, and the dorsal respiratory group. Those guys in the medulla oblongata and the pons are going to help with respiratory rate by affecting muscle contraction in those muscles that control respiration. So remember those chemoreceptors, chemical receptors in these areas are going to be measuring what? Or monitoring what constantly? pH. What is this one? Those are changes in P, partial pressure. When you see the P in front of anything, that's partial pressure of carbon dioxide and blood pH. So these are some of the different influences on the respiratory system. Higher brain centers, again, those wonderful little vagus nerves and so forth are going to help initiate different responses. If I change the amount of oxygen that's dissolved in my blood, if I use it up faster than I normally would, that's going to change the concentration of oxygen. That's going to change the pH. So they talk a little bit about exercise and altitude and how those partial pressures can influence your breathing rate. Everybody good with that? The other thing, before I get too far away from it, this diagram. We did this in lab, right? Did everybody understand what they were doing in lab when they did this? Okay. These are respiratory volumes. You should understand how or what all these numbers mean. When I'm just sitting here breathing in and out, that's a tidal volume of air. And it's about 500 milliliters. But I can breathe in way more than that, and I can breathe out way more than that. The breathe in way more than that is the inspiratory reserve volume. The breathe out way more than that is the expiratory reserve volume. And you cannot possibly, just because of the way you're made, breathe out every little bit of air that's in your lungs. There's always going to be a little bit left, and that's called residual volume. You should know how to calculate all of those different capacities with those volumes. So what are you going to need for the next test? A calculator. So if I asked you to calculate the inspiratory reserve volume. Could you calculate that? No. What would I have to do? I'd have to tell you that. But if I asked you to calculate the vital capacity, what information would you need? Yes. So you would need the IRV, inspiratory reserve volume. You'd need the tidal volume, and you'd need the ERV, or expiratory reserve volume. You with me? What you're going to get is a patient profile. There's going to be several things on it. That information has to be on it, right? Other thing would be breathing rate. What's breathing rate? How many times they breathe per minute? Does all the air you breathe in all go down for gas exchange? No. Remember, we have a respiratory zone and we have a dead zone or conduction zone. When I talk about a dead zone, what's that mean? Yeah, no gas exchange. So we're talking about the mouth, we're talking about the pharynx and the larynx and the trachea and all those bronchioles. There's no gas exchange happening there. They're just carrying stuff. Your dead zone, the amount of air you breathe in, is very closely related to your weight. So has anybody ever experienced getting heavier? What do you think is going to happen to the air that you put down in the alveoli? Are you going to be able to put as much air down there for gas exchange as you gain weight? No, because more of it will hang in your dead zone. So I put on my weight and it's like, ooh, I used to be able to walk up these stairs fairly easily, but now I need a tank of oxygen to walk up these stairs. How come? Because your dead zone is increased with your weight.
Well, the tidal volume is the same, it's just it's not using as much.
Not as much as getting down to the alveoli. So you also need to be able to calculate alveolar ventilation rate. How much is actually getting down there for gas exchange? And you're going to need to know what dead zone is. And in your patient profile, am I going to say the patient's dead zone is this? No. I'm going to say what? I'm going to put his weight in. And the other thing you have to know is how many times a minute do they breathe. So all of this information will be in the patient's chart. And you're going to have to extrapolate it to calculate these things. Okay? Then, my bone of contention at the end of the chapter. So everybody good so far with everything? Chronic obstructive pulmonary disease. And I gave a big long spiel about it in the lecture that I posted online. But I want you to do what I said to do online. Did anybody do it? No. Most of you don't even know what I'm talking about. But more than 80% of the patients that have this group of diseases, chronic obstructive pulmonary diseases, have a history of what? Smoking. That number is nauseating. All of you youngsters know how to Google stuff, yes? So I want you to go and Google what the average number of patients in hospital care to date is that fall under the category of chronic obstructive pulmonary diseases. And this includes, and you should know, emphysema, bronchitis, asthma, tuberculosis, lung cancer. They're all there. Get a number and then multiply it by 0.8. Those are all the patients that are in hospital being treated for this group of diseases that chose to be there. Yes? That's a conscious decision you make to smoke. And who's going to take care of you? We do. Yes? That's a lot. That's scary a lot. To be in the hospital and suffer. By the way, I also want you, while you're Googling this information, I want you to hold your breath. Because that's what you're going to feel like when you have these diseases.
The years is 1/3 of the world's population is infected by TB.
Scary. Yes?
That's
So that 80%, the people that chose to be there, that number is staggering. If that doesn't make you quit smoking, I don't know what will. Because you can quit smoking and heal those lungs and maybe not fall into that category when you get older. So, yes. Yeah. That's a genetic issue. So you can induce this deficiency with environmental factors, and that deficiency is going to lead to trouble breathing. So sometimes it's genetic. So we have to also think about the other 20% that don't fall into the I smoked myself to death category, right? So those are some of the factors that influence respiratory function. Remember, I have to be able to blow up my balloons and release those balloons. Anybody know anybody with cystic fibrosis? Okay, so those are things that are not under the control of that patient. They have a genetic problem with creating the proper chemicals and enzymes for respiratory function. Fibrosis of the tissue, dying of the tissue. Think of that balloon again when it's been sitting outside for way too long and it gets crusty and crispy, and then try to blow it up. So anything that's going to affect tissue that way will also throw people into this category as well. But again, 80%. Did that answer your question, by the way?
No, but that's okay.
No, which?
I wondered what antigens.
It's an actual chemical that helps with the elasticity of the connective tissue in the lungs. So if I have a genetic defect that causes a decrease in this chemical, it's going to result in problems with lung function. Did that? We good? So COPD. Developmental aspects, we're done. Any questions on chapter 22? No? All right. So all of that stuff we talked about today, I know we went through it fairly quickly, but you're going to need to know that, we're nowhere near done, so don't be zipping up and going away. Because now we have to go on to what? The digestive system. Okay, I'm going to skip forward a little bit, and the reason I'm going to skip forward a little bit is for the benefit of everybody who has to take a lab exam next week. I had a silly question from a student who asked me, well, we didn't cover the digestive system yet in lecture. Does that mean it's still going to be on the lab test? Yes, because you're going to do a lab associated with the digestive system. Who had lab on Wednesday? Nobody? You did? Did you do all your series of enzyme experiments? You did? Was it blasty fun?
It was fun.
Okay. And those experiments related to these diagrams. So in your textbook and in your lab book, you have a series of diagrams in chapter 23 that talks about the digestion of substance. On page 893, figure 23.32. The third part of this lengthy chapter talks about the physiology of digestion and absorption. So we're talking about the chemistry of breaking down large things to small things. The digestive system is for what? Why do you have one? Makes big things small. That's its job. And it's going to do that in a couple of different ways. One, mechanical. I'm going to actually take it and smush it up. And what do you do when you do that? You chew stuff. Then I'm also going to add chemicals to it to break it down. You spit all over it, saliva, and you throw it into more chemicals in the stomach, gastric juices, more chemicals as we pass through the rest of the tube. And basically the digestive system is a giant tube. It starts here and it ends here. Yes? And the whole tube's function is to make big things small. So this part of the chapter talks about the chemistry of what's going on. And for your lab exam, you have to understand a little bit about the histology, about some of the tissues associated with the digestive system. Not only do we have the tube, but we have accessory organs of the digestive system that throw stuff into the tube. Salivary glands, liver, pancreas. They're going to help with this chemical digestive process. And then for your lab exam, you have to understand what chemicals break down what. And that's what you're doing when you add all those things in the tubes. You're doing what your digestive system would do with certain chemicals or very specific enzymes. So, everybody remembers this from Chapter 2, because they were so excited when we did chemistry in Chapter 2. Remember how excited you were? And when we talked about organic molecules, we talked about 4 different groups of organic molecules. What were they? Oh, chemistry enlightened folks. Carbohydrates, proteins, lipids, nucleic acids. Those are the 4 major categories of big molecules that we have to make small during the digestive process, right? Because we want to use their building blocks to make our own. Correct? That's why you eat. You eat for building blocks. You eat for materials you need for your metabolism. So carbohydrates, when I eat carbohydrates, big carbohydrates, I want to break them down into their small units so I can use them for my chemistry. Yes? So, starch, disaccharides. What's a disaccharide? What's a monosaccharide? That's the building block of carbohydrate. Oh yes, you had to remember all that chemistry crap, because now it's going to come back to bite you. Yes? So, saccharides, monosaccharides, disaccharides, polysaccharides. We want to break them into monosaccharides so we can use them for our chemistry. Monosaccharides are things like glucose and fructose and galactose. Disaccharides, lactose, maltose, sucrose. You know this already, or you should know this already. So we're going to break the big things down into small things in the digestive system. We're going to start right in the mouth. We're going to add some chemicals called amylase. And amylase is going to break down carbohydrates. It's not going to last very long because it needs a nice neutral-ish pH to work. And what happens when I swallow food and throw it into the stomach? Woo! Acid bath. Yeah. So it's going to get neutralized. But the pancreas, thank goodness, will pick up in the small intestine as I move through the tube and add chemicals. And so will the small intestine itself. Because when we look at the physiology and anatomy of the small intestine, we'll see that the cells that line it actually make some of these chemicals too. So we see here under carbohydrate digestion, and I know I'm putting the cart before the horse, but I want to just at least have you hear this before you take your lab exam next week. And if you had lab on Tuesday, it's going to be the following week. that we have chemicals that are secreted from the mouth, the small intestine, and the pancreas, which secretes into the small intestine, that are going to help break down big carbohydrates into small carbohydrates. Then what do we do once we get them small? How do we use them? We have to get them where? Into our transport system. We absorb them. Most of the absorption will take place of these smaller things now in the small intestine as well. So those little small intestine cells are very, very busy. Not only are they making chemicals to help me continue to break things down, but they're also going to help me with absorption out of the tube and into the circulatory system. You with me? The other major molecule, of course you know this, is protein. These are organic molecules, which means what? They contain carbon. So proteins are made of what? What's their monomer? What's their building block? No, peptides are the bonds that hold them together. Amino acids. How many different amino acids do we have in our bodies? 20. And combinations of those 20 create our proteins, big molecules. So when I eat a cheeseburger, you're not a cheeseburger, are you? Most of us are not cheeseburgers, right? But we need, as far as the protein part of the cheeseburger goes, we need to break it down, break those amino acids apart, break the peptide bonds that hold them together, and then use them to do what? Make our own proteins. So the digestive process is going to allow me to do that. So I eat the cheeseburger, and in the stomach, that nice acid environment, we're going to talk about some of the different chemicals produced by the cells there, but they're going to help me break down proteins. Pepsin, we'll talk about next class. The pancreas is also going to kick in some chemicals in the small intestine. Trypsin, chymotrypsin, and carboxypeptidases. Chemicals that are going to help break peptide bonds and give me free amino acids. And then, of course, the lovely little small intestine is also going to give us more chemicals that are going to help with this process. What else is going to happen here? Once we hit the small intestine, everything hopefully eventually will be small enough for what? Absorption. So amino acid levels, amino acids leave the epithelial cells by facilitated diffusion. What's that? The cells are going to help grab onto stuff and spit it out on the other side. See, all of this stuff you had to know. And who's going to help me transport all of these things? Yeah, circulatory system. But we're going to see that the liver is a very busy little organ. We've talked about it a couple of times already. But the liver is sort of the bypass before everybody else gets their goodies. So when I absorb things through the small intestine, it has to go through the liver via what we call the hepatic portal system before it gets distributed throughout the body. So that's proteins. Who else? Lipids, the other organic molecule you all told me about earlier. Fat digestion. What are fats made of? Go back to chapter 2. Fatty acids and glycerol. Yes? Back to the cheeseburger. Is that fat the same as your fat? It's kind of close, but what do you have to do in the digestive process? Rip it apart and put it back together again. So we have some chemicals that I'm going to spit all over them right in the mouth. Lipase is going to help me with fat digestion. Again, it's not going to last too long. The gastric stomach is also going to make some lipase as well. And then the liver is going to help, and the small intestine. you know what a gallbladder is? What's a gallbladder? They talk a little bit about that here. It stores a chemical that's going to help me with fat digestion. The chemical's called bile, but it's just a storage and concentration place. Who actually makes the bile? The liver. Way back when liquid detergent came out, none of you remember this, and you can still buy it like that. It comes in big, huge bottles, right? Big honking bottles. And we used to have to use like a cup of it to wash our clothes. But nowadays, how much do you use to wash your clothes? Just a smidge. Is it different than what we used to make back then? No, it's the same thing. But what did we finally figure out? Instead of having to carry around these big honking bottles, what did we figure out would work better? Yeah, if I suck a lot of that extra water out, you can still get the same amount of soap, but now in a much smaller space, right? It's concentrated. It's still going to have the same effect, and that's why you're not supposed to put a lot of soap in your clothes. Can actually work the opposite. Just because your clothes are really dirty doesn't mean you should use more soap. It'll actually get caught up in your clothes and work the opposite. Did you know that? Yeah. So, bile production is kind of like that soap I described. It's very watery. We send it to the gallbladder where we're going to concentrate it, so that when we actually squirt it out into the small intestine, it's like that soap that we use that's concentrated for our clothes. That's going to help with fat digestion. When we talk about lipids, what was one of the things we talked about when we talked about that molecule? It's not bipolar. It's hydrophobic. It's afraid of the water, right? So when you take in lipids, they tend to, and pretend each of my fists is a lipid molecule, they tend to hook together. Remember, they're made of glycerol and fatty acids, right? I need to unhook them before I break them apart. So bile is an emulsifier. It doesn't break them apart, it just pulls their hold on each other. Takes them away from each other. The lipase, the enzyme, is actually what breaks them into glycerol and fatty acids. So we have extra steps for fat digestion. First, we have to add an emulsifier, bile, that's going to break their hold on each other. And then we add the enzyme, lipase, which is going to break them apart into glycerol and fatty acids. They're still really big, and they have to be transported. And we'll talk about this in detail next class, but at least I want you to hear it before you sit for your lab exam. We create little carriers of our lipids. And because they're so large, we actually can't absorb them directly into the circulatory system because they're too big. Yeah, remember when we talked about the lymphatic system? One of its jobs was to help with lipid absorption. So when we look in the anatomy of the small intestine, what we're going to see is not only little blood vessels inside all of these little finger-like projections, but we're also going to see lymphatic vessels as well. And they're going to help with the absorption of lipids into the circulatory system. But I can't go directly there. I have to bypass and go where? Through the lymphatic system before I get dumped off. And what's this? Where's the thoracic duct dump? Remember? We talked about the lymphatic system? It's going to dump into the circulatory system. So I have to bypass because I'm too big to get directly into the circulatory system. And we'll talk a little bit about that chemistry in more detail. So we're going to get lipids into the circulatory system eventually with the help of emulsifiers and with the help of enzymes. And then last but not least, we have nucleic acids. Where do I find nucleic acids? DNA, RNA, deoxyribonucleic acid, ribonucleic acid. Where do they come from in the foods that you eat? Everything that has a nucleus has that stuff. What are they made of? We have a sugar, we have a phosphate, we have a three things. Yeah, nitrogenous base. Remember that? Those three little things together make up nucleotides. And the order in which they're all stuck together are your genetic information, yes? Is your genetic information the same as a cheeseburger's? No. Nope. So what do you have to do with the cheeseburger? Break it down so that you can then have free nucleotides. Remember we talked about free nucleotides in the nucleus and in the cytoplasm during a process called transcription and translation? That ringing a bell? Chapter 3. Remember that? So how do we get the free nucleotides? Like this. So we eat them, break them apart with the help of who? The pancreas. And we have deoxyribonuclease enzyme that's going to help us break it apart, and nucleases that are going to help break it apart, along with enzymes in the small intestine. And you'll hear that group of cells that line the small intestine referred to sometimes as the brush border. Because when I look at them under the microscope, the cells, if my hand was a cell, have these little extensions on the top. They're called microvilli, and they increase surface area, but when I look at them under the microscope, they look like a brush. So the cells are called the brush border. And all of the chemicals that they produce are called brush border enzymes. Yes? That's easy, right? So for your lab exam, and that's why I wanted to do this first, you need to know that diagram. You need to know the 4 different organic molecules. You need to know their building blocks. You need to understand who's involved in breaking them down. Okay? So the enzymes that are involved in breaking them down. That's going to be on your lab exam. So I'm just giving you a heads up. We good? Just a quick look. Remember, I'm going to take small things, bring them into the circulatory system. So this is a quick look, and we'll go back to it, but I just want to show you quick. These are the cells that are absorbing all of those materials from the tube that we just broke down. Remember I told you that brush border? These are the cells that line who? The small intestine. See, they kind of look like a little brush head at the top. So this is some of the chemistry involved in actually taking what's in the tube that I've now broken down and made small and bringing it into the circulatory system. So these are some of the molecules or protein carriers that are involved in endocytosis and exocytosis in the small intestine. See, all of this stuff is from first semester. Yes? Everything builds on itself in this course. These are the little units that were mentioned under lipid digestion. And with the help of bile salts that pull those clumps away from each other, we can create these little packages called micelles. And their micelles are made up of what? Fatty acids and monoglycerides. Because remember, this is a hydrophobic molecule, so I need help what? I need help transporting in that liquid environment. And that's what that little package is going to do for me. It's going to allow the cells to pick it up, that brush border, and dump it out, but it's not going to go into the circulatory system directly. It's going to go where? What's the little green thing down there? It's going to go into the lymphatic system, and it's going to then form a complex molecule called a chylomicron, basically a carrier for lipids.
So it's salt to break down lipids?
The salts, the bile salts, aren't going to break them down. They're going to break them apart. The enzymes later on are going to act on them to break them apart. Lipase. Yes? Does that make sense? Okay, so I want to show you that before, and I have 5 more minutes to talk, and I'm going to talk for 5 more minutes. So, I'm good at it. Digestive system, big tube, beginning to end. Beginning starts in the mouth, falls down into the next part of the tube. This is a tube, and the tube will change as it goes along, but it's just a big tube. The tube starts at the esophagus. We're going to throw food down into the esophagus, and then the tube is going to fatten up a little bit in an area called the stomach. It's going to continue along this tube to the small intestine. Now, if I stood here and pulled my small intestine out and threw it across the stage, it would hurt, and it would probably bang into that column right there. How long is that part of the tube? About 21 feet of small intestine. Then the tube's going to fatten up and change a little bit as we go along into what we call the large intestine. Less of that, about 12-ish feet, 12 to 14. You look in 100 different books, you're going to find 100 different numbers. But it's ish, okay? So 21-ish, 12-ish feet, different for everybody, right? Of large intestine. And then we're going to dump out rectum, anus. That's the tube. Now, all the other stuff that we're going to talk about with respect to the tube, we're going to refer to as accessory organs to the digestive system. So in this diagram, who are we talking about? Well, we're talking about the tongue. We have glands that are going to shoot stuff in there, salivary glands. We have this guy, the liver, and the gallbladder. Why do you think there's little stars beside all their names? Diagram. They're the accessory organs. Yes? So salivary glands, we have a whole bunch. And they all surround this oral or buccal cavity. And they squirt into it. Does anybody know what a gleek is?
Yeah.
Sometimes somebody's describing something to you like delicious hermit cookies in the bookstore. And sometimes you start building up all this saliva. Because believe it or not, your brain is involved in sending messages to your salivary glands to salivate and get ready for food. So they're there. Pancreas. Spleen is there, but he's just I don't know why they put him there. He's not part of the digestive system, but he's there. Hi, spleen. And then these are different parts of the large intestine, and these are different parts of the small intestine. So we will pick up, wait a minute, I got to ask a clicker question, so let's see if we can. No. All right, let's see if we can do this, just so we can take attendance. But all right, the esophagus is part of the what, while the pancreas is part or considered what when it comes to the digestive system. And I just said it, so if you don't get it right, I'm going to kick your butts. By the way, another name for the digestive system is the alimentary canal, or the GI tract. Just a hint. Did everybody click? 5, 4, 3, 2, 1. What's the answer? A is the answer. Yes! All right, my friends, depart. We'll pick up, again, if you have any specific questions on the respiratory system that you were too afraid to ask in class, make sure you bring them with you on Tuesday, okay? Because guess what? After this stuff, we get another test coming up.